Okay. So, welcome everyone. My name is Juana Manuel, and I'm a second year PhD student in Dr. Heather Mefford's lab. I have the pleasure to introduce Dr. Kaitlin Samocha. Dr. Samocha joined Massachusetts General Hospital in 2021 and Harvard Medical School in 2024 as an assistant professor. Prior to this, Dr. Samocha did her PhD in genetics and genomics at Harvard University under the supervision of Mark Daly and completed her postdoc in Dr. Matthew Hurl's lab of rare disease genomics at Wellcome Sanger Institute. And her research interests focus on developing methods and statistical tools to improve interpretation of genetic variants, particularly rare variation. Dr. Samocha has created a mutational model to predict the expected number of newly arising variants that have helped identify dozens of disease-associated genes for autism spectrum disorder, developmental disorder, and many others. Dr. Samocha is on the NOMAD committee and is part of the Translational Genomics Group at the Broad Institute, which has a mission to support the discovery of genetic basis of rare disease and translate their work into medical practice by focusing on community-centered projects that promote collaboration, data sharing, and open science. In 2015, she received the Charles J. Epstein Trainee Award for Excellence, or it is now called ASHG Trainee Research Excellence Award in Human Genetics Research. Today, she will be presenting on using large genomic data set to improve variant interpretation. Thank you, Dr. Samocha, for joining us, and now I hand it to you.
Good. Yeah, I have pause places. Yeah, I'm totally happy to be interrupted. I have 4 vignettes that we'll go through and we can expand and contract as needed based on time and people's hunger level. So I'm happy to adjust on the fly as we go. So I'm very excited to be here with you all at UT. I was very curious to see this giant pyramid, learned it was a Bass Pro Shop, really cool, like fascinating things. But we're going to be focusing on other fascinating things, which is the large genomic data sets, which are publicly available, and how we can make best use of those. So here we go. So I'm not going to belabor the point too much, but one of the major goals in medical genetics is being able to identify genomic variation that is influencing a trait or disease that you're interested in. This is a figure from 1,000 Genomes Project from 2015, and we can see for populations around the world, there are about 4 to 5 million genetic variants per genome, with European populations, genetic ancestry populations over the left on the 4 million side, and African genetic ancestry populations on the left, on the right, with a more closer to 5 million. However, the tricky thing is now not identifying the variation necessarily, but interpreting it. And that's because of all of these millions of genetic variants, the vast majority of which are going to be neutral or fairly benign when it comes to a particular trait or disease. So I'm not the first to say this, I won't be the last to say this, but if you care about interpreting the genetic variation you see in an individual, particularly in a rare disease setting, we really want to study the variation on a population level. So you can use the patterns of where variation are occurring or not occurring to help us interpret what we see. So we need to establish some useful ways of prioritizing potentially damaging variants when we have, for example, a patient in a rare disease setting. I am not a medical doctor, but phenotype is incredibly important. If you are in the clinic and you are trying to make new disease gene phenotype association, you need phenotypic information. So I just want to put that up there to say that is incredibly important. But we're going to focus on the genetic level evidence you can use. So there's both variant level pieces of evidence, for example, functional consequence, loss of function, usually having a larger impact than a synonymous variant. There's also a number of in silico deleteriousness predictors, or some of these functional assays that are coming out, these MAVE data, where they test every single type of change. And what we'll talk about a little bit here is the presence or frequency of a variant in a database. On the gene level, you can also use, for example, prior association to disease. That's a great piece of information to keep in mind, or expression. However, we'll argue again here that if a gene is intolerant to being mutated in the general population, it might be important in rare disease cases. So this is an excellent time to be doing human genomics. And some of that is because we have access to increasingly large sequencing data sets of reference populations, and these are publicly available or easy to access. So when I used to show this slide, there were a lot more data sets on there, and I've just cut it down to those that are over 100,000 individuals. So we have on the left the top med, so Transomics for Precision Medicine. Their Freeze 8 was 132,000. We have the Genome Aggregation Database, or GNOMAD v2, was 141. All of us is 245, but they're about to release over 400. UK Biobank floating a little under 500,000. And then GNOMAD's most recent release and the Regeneron Genetics Center Million Exomes dataset are both a little over 800,000 exome or genome sequenced individuals. Now, these are all being colored here by inferred genetic ancestry group. And none of these databases are globally representative at all. And I really want to emphasize that point. This is genetic similarity to a labeled group. It's nothing to say about identity at all. It's just saying you cluster with individuals that have been labeled as coming from a particular place. But there is a lot of this light blue color all over these bars, indicating broadly defined European genetic ancestry. So if you're coming from a population that's not well represented, or your patient is coming from a population that's not well represented, you are going to have potentially less value out of some of these databases. We still think sample size is a really big deal, but there are some population-specific variation that might be missed. So in particular, I want to highlight Middle Eastern, this kind of dark blue. There's a couple pixels here and here for it, but that's not a lot. It's not globally representative. And Oceania isn't on here at all. We're working on it, but this is a thing to keep in mind. And it does matter. So this is, I'm associated with a project called NeuroDev, which is looking at developmental disorder cases from Kenya and South Africa. And both of these are populations that are not well represented in GNOMAD because it's East and South African genetic ancestry, where most of the African American samples are coming from West genetic African ancestry. So we can see if we look at rare variants, so this is a minor allele frequency, less than 0.1% across populations. These this purple color is coming from the African American ancestry populations in GNOMAD. And these other two, kind of like this pink and this other purple, are coming from Kenyan and South Africa from NeuroDev, and they have far higher number of total or unique variants than what we see in our population. And that's filtered based on our population. So in total, this is one way that you can start to limit the number of candidate variants you're looking at. Allele frequency. So you're already dropping down to only 10 predicted loss of function variants, 200 missense variants, et cetera. And that variant function information is also another way to filter, but this is a major impact here. So these are the 4 vignettes we're going to go through, time permitting. And if we want to pause at any point, happy to do that. So the first 3 are coming from the GNOMAD dataset itself. So our most recent release, 4.1, had 731,000 exomes and 76,000 genomes. A lot of our resources are slowly being updated from V2 or V3 to V4. We take a lot of time to make sure our data sets are cleaned and quality controlled before we redo all of our research analyses on them. And the last one is actually not quite related to GNOMAD, but it's about automated reanalysis for rare disease diagnosis. So focusing first on variation, one of the most powerful ways to use these databases in a rare disease setting is to remove variants that are too common, right? If we went back to this graph here, you want to get, if you're studying a rare disease patient, you want to filter out anything that's been seen in a lot of people. It's probably not causing their rare disease. So you can use allele frequency information in these populations, and you can use these predicted loss of function, peel-off, missense, indels, that kind of information. However, we can do a little bit more in this space. The first thing that I'll mention briefly is refining these allele frequencies estimates using local ancestry inference. So from genomes, we can take our admixed populations and actually find the continental background of genetic variants themselves. So if we look, for example, at admixed American populations, it's coming up from a 3-way admixture of European, African, and continental American or Amerindigenous backgrounds. And so each individual actually is a combination, like a patchwork across their genomes. So Mike Wilson on the GNOMAD team did this for the admixed American. We've also done it for African and African American. But what you see is in this group of admixed Americans that you have some people that have fairly homogeneous backgrounds, mostly coming from one, but we also see both 2- and 3-way admixture in these individuals. And if you can start to paint the chromosomes based on their their continental ancestry track, you can get much more refined allele frequency estimates. So the example I'll show here is coming from COL27A1. So this variants in this gene are associated with Steele syndrome. And there was a theory that there was a Puerto Rican founder variant. So one of the deleterious pathogenic variants in this gene was seen at a higher rate in the Puerto Rican ancestry set. And if we look across the nomad populations, it's pretty rare globally. We see a much higher allele frequency specifically in the admixed American populations. But when we go in and apply our local ancestry inference, all of that is actually coming from the continental American or Amerindigenous background. The local ancestry inference from African or European genetic ancestry groups is much lower and matches the global. So this is one of those places where the background matters and can help us refine allele frequency estimates. This is mostly a way to rule out variants, right? This is potentially too common. This one actually supports that it was a Puerto Rican founder event, which is nice genetic evidence here. So this is, we're going to move swiftly on from this, but this is one thing that you can do when you have genome data. However, I really want to emphasize that the more interesting something looks, the more likely it is to be false. So if you come to our data set or any other data set, UK Biobank, all of us, big, and you see something that looks really cool and interesting, it is your job to prove that it's not. And if you cannot prove that it's not interesting, then maybe you have something right. So we spend a lot of our time trying to quality control this data set, but things still slip through. So for example, if you look in the Nomad data set, you will find loss of function variants in genes that you might not expect to see them in. So my colleague Anna Donna-Luria a couple of years ago developed a curation framework to take loss of function variants, so things that are nonsense, splice site, frame shifting indels, and to evaluate whether they're actually seem like they're causing a loss of function. So they did manual curation of all homozygous loss of function variants in the V2 data set. They found about 28% of them look like they're not actually loss of functions. Okay, so dropping about 28%. Similarly, in 22 recessive disease genes, they looked at the heterozygous loss of functions and found a very similar rate, 27, 28%, that also seemed like they weren't loss of functions. We have these curation results up on our Nomad browser, and you can get some information about why we don't think it's a loss of function variant. So for example, it's in a homopolymer run. There's some mapping issues. We think that it's just a dodgy call. You can also do this yourself with read data on our data set, but we wanted to expand this and apply it to more variants. And the reason is because we are seeing ClinVar pathogenic variants, things that are associated with rare and severe disease in our data set. So in Nomad V2, for example, we found 16% of all pathogenic or likely pathogenic variants in ClinVar were also in the Nomad V2 data set, and that's jumped up to 30% in Nomad V4. This trend is, it's nice to see that the benign or likely benign variants, this is what we'd expect to see, right? If it's not causing disease, we can see it in the general population. So it was a little bit of a surprise that we saw this jump up for these deleterious variant classes. And variants of uncertain significance are always a little bit of a mixed bag. Thankfully, most of the pathogenic variants have lower variant allele frequencies in V4 compared to V2, meaning we got a little unlucky in V2. We saw them in a person, but they're rarer as we look at more and more individuals. But we wanted to dig more into this. Unsurprisingly, most of the pathogenic alleles that individuals in Nomad carry are associated with autosomal recessive conditions as opposed to dominant. So everyone has, on average, about 10 pathogenic or likely pathogenic variants from ClinVar in this data set. But when you compare the pathogenic, likely pathogenic breakdown in ClinVar versus what we see in Nomad, there's a massive enrichment here of this yellow, which is autosomal recessive. We do, however, have, yeah, go ahead.
What's the minor allele frequency that you're calling?
This is across everything, but you're right. The pathogenic variants tend to be much more rare, but this is down to singletons in our data set. Yeah, I didn't pull the figure, but in the paper, which is cited on the next slide, we have a breakdown of the pathogenic versus benign allele frequencies, and the benign variants have much higher allele frequencies in our data set. A lot of the pathogenic ones are super rare, like 5 or less copies. But on that point, we do see several pathogenic variants in autosomal dominant genes associated with severe pediatric conditions in our data set. KMT2D is associated with Kabuki syndrome. We have 13 variants, 41 alleles. A lot of these are coming in from biobanks, and this is kind of a complication these days. Incomplete penetrance is one aspect of this. Or if someone is healthy enough to make it into a biobank, they're making it into our data set. One thing that was pointed out to us is we have some cystic fibrosis delta F508 homozygotes in Nomad V4, which we didn't have in V2. So this is associated with, like, the delta F508 is a classic cystic fibrosis variant, and the homozygotes probably all have cystic fibrosis, but they were healthy enough to get into a biobank, and they're healthy enough to now get into our data sets. So please keep that in mind, that any of these population resources, we call them reference populations because they're supposed to reflect the general population. Everyone has something. Like, we're not checking people's cholesterol levels to get into here, we have some like schizophrenia in here. On our website, we have a list of some of the conditions. None of these are necessarily that far enriched above their population prevalences. We do get rid of cohorts that are known to have rare, severe pediatric conditions, but you can see a few things do filter in, again, from the biobanks because we're not allowed to use their phenotype information. Right, okay, but we wanted to do a deeper dive here. So this is a paper that came out in 2025. We were looking at 77 haploinsufficient genes that were associated with severe early onset disease. So this is a set of genes that we should not see loss of function variants in our data set. But in our 807,000 individuals, we had over 4,000 loss of function variants. If we do a first filtering basis using this lofty tool to identify those that seem to be high confidence, drops it down by a little bit. So we're still talking about 4,000 loss of function variants that we wouldn't anticipate to be there. We then, I should say, Sana Goodmanson focused on the genomes because they had a higher rate of these variants, and really did a deep dive into curating these 734 loss of function variants. So again, this is applying that curation framework that I mentioned before, using secondary sequence properties, biological relevance, potential technical artifacts. And what she found was that 70% of those 734 loss of function variants had evidence that they weren't actually a loss of function. So we saw something interesting in the data set, but they're not actually real, right? So as a comparison, these are those autosomal recessive and homozygous variants I mentioned a couple of slides ago. Here's that 27, 29% that don't look like they're actual loss of functions. And this said it's 70%. Now, the vast majority of them are coming from variants that fall in the last exon, that look like they're in exons that aren't always spliced in, that have a nearby frame restoring indel, for example. So this is requiring a lot of like deep dive into this. We did notice that if there was a ClinVar variant that was considered pathogenic, it was more likely to look like a sequencing error. The benign variants were some of these ones like in a last exon or low PEXT. And then a big chunk of them had a number of different pieces of information that contributed to us saying that they didn't seem like they were loss of function. Unsurprisingly, the ones that we thought didn't look like loss of functions had a higher allele frequency. So that is a very powerful piece of information to keep in mind. This is a significant trend across the ones that we said weren't loss of function, uncertain versus unexplained. And we looked up, I think it was about 100 ClinVar variants that were being curated in this set. And unsurprisingly, the benign variants were mostly explained, and the pathogenic, likely pathogenic, had more of that mix of we couldn't understand why we couldn't eliminate them with this curation framework. Right, okay, so Sana and team members took it down from 734 to 137 loss of function variants in these genes that are unexpected. Then did a couple of other steps. So one of the first ones was we noticed that the 1,000 genomes project samples we have in here actually had a much higher rate. They're about 5% of our genomes, but they had 25% of these variants. The 1,000 genome samples were coming from cell culture. So we think actually some of these might have arisen in cell culture. So they're not real germline variants. So we dropped that data set and we get rid of a bunch. The second thing along that line of somatic mutations is we got rid of or I should say Sana got rid of those that had low allelic balance or were occurring in genes that have been associated with clonal hematopoiesis. ASXL1 is a classic one here. We see a lot of loss of functions in ASXL1 in the Nomad dataset. They're all somatic. They're not germline. That's associated with a pretty severe condition. And then the last step was focusing only on loss of function variants found in the main select transcript. The logic here was Main select had a whole bunch of curation going into it. If it's falling outside of this, maybe it's less biologically relevant. So at the end of the day, only 33 of these 734 variants remained without a plausible explanation. So we took our data set and we were able to say, what a surprisingly high rate of loss of function variants in these really intolerant genes associated with severe disease. And we could explain the vast majority of them with some manual curation and some digging into different cohorts. So that is a pause point, but we can shift to something else. Yeah. Which one?
What about the 33 variants?
The 33. Yeah, so we know which ones they are. They're listed somewhere. It is curious. There is still ideas about incomplete penetrance in the general population based on like other variants in the gene or other things in your genetic background, or maybe these individuals have some subthreshold phenotype that means that they're still okay to get into like UK Biobank. Actually, UK, there was a nice study by Caroline Bright looking at developmental disorder associated like pathogenic variants in UK Biobank, and you see a shift in like educational attainment and a number of other related factors. So it could also be that there's some leakiness with this.
So,
Yeah.
Well, we could do the questions, so, and these are not tolerant to copy number change, like you can't have additional copies of these genes because you need to make that.
So
Because then, I mean, you have a second copy.
that's actually, yeah.
Gene wide this time.
We have seen that things that are intolerant of loss tend to also be intolerant of gain. But I'll get rid of this. I have anecdotally, we have noticed when looking at some copy number variants, we think at least one of these might be that someone had a duplication of a gene and then a loss in one of them.
So at least 1/3.
Yes, yeah. So I was like, we're in super hand wavy, single
Okay.
thing, but yeah, right. So it could be stuff like that. We could do an even deeper dive into some of that information. I do expect we'd never be able to get it down to 0 because
It's happening.
Yeah.
Probably, you should see it across 33 cases. You should be able to tell.
Yep.
Just looking at it.
Yep, I think we can do a better job of that. The people that do the copy number and the people that do the single nucleotide, we could do better crosstalk. I think the copy number calls came later than the single variant calls. And so part of the reason this is on the genomes as opposed to the exomes is we started this project a long time ago, and the genomes were from the V3 release were what we had available. Yeah.
Two quick questions.
Yeah.
How do you handle X chromosome? Because you know that's going to be under a very different selection.
So in this curation, we can talk about it when we get to selective constraint. In this curation framework, they have different considerations about what they might expect to see, but it doesn't, I don't think there are, I actually don't know if there are X specific rules. But I guess you'd expect male versus female, so that would come into play there.
And the second question is, how do you, so pathogenic or variant depends on the age of onset. Since you're interested in neurodevelopment, sure. You know, you could say age of onset before 20.
Yeah.
As your disease criteria. For
Yep.
prevention. If it's an ApoE4 allele.
Yes.
What do you do with it?
That's why-
It's a Huntington's allele.
Yeah.
What do you do with it? Because it's actually beneficial in terms of fitness early.
That's why we did this on 77 haploinsufficient genes associated with severe early onset disease. Because you're right, anything that's associated with late onset, we do and we expect to see variants in our data set. Our average age is about 50 at time of collection. So there are plenty of people in there that could go on to develop cancer or Alzheimer's or things like that. We'll talk about actually selective constraint. I'm focusing on missense variants, but we see like BRCA1 seems actually fairly tolerant of loss of function variants. It's not completely depleted. And that's because it happens after reproductive age. That's typically after onset is, yeah.
There's no way to track health records and so that's
Sometimes, so since we pulled in UK Biobank, if we know they're a UK Biobank sample, we might be able to go back and track them down. We have a really tight IRB. We're not allowed to use phenotype information at all. This is like, we got this IRB, actually, Daniel MacArthur got this IRB back a decade or something ago. And we're allowed to do all of this aggregation and release because it's aggregation, because there's no phenotype. Yep. No, it's a very good question. And that's why often, like if we see these 33 and we see they're in UK Biobank or 1,000 genomes or something where you'd, actually, I don't know if you have phenotype 1,000 genomes. Like if we can go track them down somewhere else. Yeah, right,
It's not different between items. It's probably a bit of
right.
if you had some of those genes, it's probably different.
Yeah. But
But our view is here, which is fun.
anyway, yeah, well,
Phenotypes are not.
right. But you're right.
Caitlin, here would be possible as without a neutralizing explanation.
Yes, yeah. So, and I guess I should say that some of the, like a loss of function variant falling in the last exon doesn't mean it's not pathogenic. There's a whole bunch of genes that have a mechanism that is nonsense mediated decay escape. Like MM1 has a very particular neurodevelopmental phenotype for loss of functions that only happen that last exon. This is a large swath sweep. I mean, honestly, it takes a long time to do manual curation of this, which is why we did it on 734, which is like a heavy lift. We're interested in training like a machine learning model to maybe give like a probability of something being real or not based on some of these characteristics. But things like frame restoring indels, like there aren't that many callers that are aware of what's in the general vicinity, and you can try to scan through sometimes. If it's nearby, super nearby, you can say, oh, this is in cis, this is a frame restoring indel. But if it's farther away, it gets more complicated. So there is a little bit of manual aspect here that may be hard to replace fully.
It's funny the variant colors use. It's in the reads.
It is in the reads. Well, yeah, but this has been the case for decades, right? GATK doesn't do this.
Just because
Yeah, I know. I mean, if you have a caller that does
yeah,
this,
you do.
okay.
Yeah,
Wow.
in fact, we used it in cell calls in
Okay.
the House Genomes Project, and they were this is actually in response to stuff that they garbage showed. It's even the exome. It's like the one that wants something.
But you can do that when they're nearby each other, though.
Yeah, so I mean, onto a read line.
Yeah, okay, 150 base pairs.
You know, loss of function by the indel, but they're all restored.
Yep.
If you percent it, 10%.
Yep.
Yeah, but I did that. I checked it out as a being. Regardless.
Mhm.
Excellent.
Yeah.
Shows.
Okay. We should, this is doable. You make a good point. This is doable.
We should be successful with GATK.
Yeah.
I'm really surprised.
It's not a standard thing in human genomics. I mean, we'll talk about that a little bit if we get to the phasing thing. Do you want me to just start talking about this one?
You should just go over.
Yeah.
Okay, no, that's fine. No, as I said, I can expand contract. Right, so I feel like I can't give a presentation without talking about the selective constraint. And this is mostly that the nice thing about these reference populations is identifying the like patterns of variation, so where they occur. And there's been two primary approaches here to determine intolerance to mutation. So the first is a shift in the allele frequency spectrum, so more rare variants than common. Slavoj Petrovsky and David Goldstein had this RVIS approach that did that. Our approach has been focusing more on a depletion of rare variants compared to expectation. So the logic here is if you have a tolerant gene on the left and a constrained or intolerant gene on the right, mutations arise, potentially they get selected out. And at the end of the day, if you collapse individuals in the population together, you see a number of rare differences in between individuals in a tolerant gene, but a constrained gene is lacking that expected amount of variation. So we did have to make a mutational model to do this, to prove that it works well. I'm showing you on the x-axis all rare variants expected from the model, and on the y, all rare variants observed. And when we do this on a gene level for synonymous variants, we see incredibly high correlation. So 0.93 for the vast majority of genes, we can predict synonymous variants well, which means that we trust that we can also predict missense and loss of function. The logic here, the idea is since our model is selection neutral, any depletions we see away from expectation should be due to the action of selection. There are always going to be some outliers here, some technical reasons that might not be the case, but in particular, if we look at loss of function variants, almost all genes have less loss of function variants than we'd predict based solely on their sequence. So we've come up with a number of different scores to quantify this. An older one that's dichotomous is PLI, or the probability of being loss of function intolerant. If it was, this came from an EM algorithm, if you're above 0.9, we said there's about 3,000 genes there that are incredibly intolerant to being loss of function, of having a loss of function variant. And the more recent one is called LUF, or the loss of function observed over expected upper bound fraction, which is a continuous metric. It's really taking the observed expected we seen in the data set and putting some statistical bounds around it, because we know that this is a sample of the population. And again, the lower this is, the more constrained it is. We've been able to show that these loss of function metrics help when we're trying to prioritize potentially pathogenic variants in cases with severe developmental disorders. It lines up with what we'd anticipate to see. But I wanted to focus actually on missense variants a little bit here. And that's because missense variants are far more challenging to deal with, because it depends on the specific amino acid substitution and where it occurs in the protein. We also know that there are many more missense mutations per exome. So loss of functions are more rare. Like you have fewer opportunities to create a loss of function than you do a missense mutation. And the last is that there are a lot of variants of uncertain significance that are missense mutations. And we do see from developmental disorder cases that there is clustering of missense mutations in certain domains or regions. So we don't actually think it's the whole gene necessarily that's missense intolerant, but probably only sections of it. So there are a number of different ways to go after this. One way is linear. So you can look in genes like DNA space. This is constrained coding regions. This is the approach we've also taken. So you say these exons here are depleted. There's also more of a vertical approach, looking at homologous regions or domains across different members of the same family. And especially with AlphaFold 2 coming out, structural or 3D. So looking at like 3D balls around particular amino acids to find what's going on. So we've done both the first and we're working on the last one. I'm going to talk about the approach we've had. So this is being actively updated to the most recent Nomad dataset. It always takes a while to move our research projects over. Our approach is one of these linear ones, and we're trying to find ways to break transcripts into regions with significantly different tolerance to missense variants particularly. So in this play example here, let's say the first half of the gene has 80% of its expected missense variation. That's fairly tolerant. And the end half has only 20%. I should say we have a bioRxiv preprint up on this. We're updating it, and hopefully the new one's out in a couple of months. Again, it takes a while to move everything to a new genome build and everything. So our approach here is we start with canonical transcripts, and we have a recursive approach in base pair resolution. So we're sliding through doing sort of like nested likelihood ratio tests to say, is this a way to split a transcript up into different regions? And just to summarize it, about 30% of all transcripts have evidence of being split into multiple regions. So you might think, well, why not more? When we run it in V4, it jumps up to about 35%. But there's probably a cap here. I don't think everything's going to get split. We are seeing a median region length of about 460 base pairs that is dropping in V4 because we have more power. And we do see that the local missense observed over expected ratio can be much lower in these regions than on the gene level or the transcript level itself. So we are able to actually refine the local missense constraint for this. And I think it impacts about half of all coding base pairs. Re-annotates that. To check if this is actually pulling out interesting signal, the first thing we do is we compare the missense observed over expected values, so these bins here of all coding base pairs, to the de novo missense variants that we find in controls. So these should be falling. These are control individuals. They're actually the neurotypical siblings of autistic individuals. So the idea is that if de novo variants are falling as mutations, indicate they should match the overall distribution that we have here, and that's exactly what we see. As we shift to ClinVar pathogenic and likely pathogenic in these severe haploinsufficient genes, we see a massive enrichment of those in the most depleted missense regions in the genome. And kind of in between, if we look at de novo missense variants from developmental disorder cases, they're in between the two of those. And the reason that's the case is because not all de novo missense mutations in a developmental disorder cohort are going to be pathogenic. It's a mix of damaging and benign. So they are somewhere in between the two. So we did also want to just check a couple of things. So we were looking at per variant heritability. So moving a little bit out of the rare disease space, we were looking at common protein coding SNPs in these observed expected quintiles and looking at enrichment from linkage to disequilibrium score regression. So just mostly to say that the most depleted quintile here, these most depleted regions for missense variants in the exome are also enriched for common variant heritability. And there's some clinical people in this audience. If you work in a clinic, there is calibration to determine the level of evidence that you can assign to particular classes of evidence to determine pathogenicity. There's this ACMG framework. So you say a de novo variant gets a certain amount of weight to say it's pathogenic or benign. If it's seen in a population, that's evidence for it to be benign. So we did clinical calibration here on this. And since this isn't a super clinical audience, the short version I'll say is that the depletion regions that we have can actually be moderate evidence that something is pathogenic. And that's very similar to both another regional missense constraint approach, as well as this COSMIS, which is a 3D constraint approach. So all of this is saying depletion in the general population can actually be used as evidence that something is pathogenic in a rare disease case. Not right now. This is right. So we do this on all of the samples together because that gives us the most power. If we were to start to cut it down into different groups, we'd lose power. Yeah. On the loss of function per gene level, we can potentially start doing that now because our cohort's getting big enough, but we really want to like zoom in on refined regions. It gets more challenging. Okay. Another breakpoint. Any questions about selective constraint? I didn't talk about the gene level stuff much, but I can answer questions about that. Cool. Let's talk about phasing. Oh, did you have a question? No, good. Right. So the other way that you can use patterns of mutation is not just the depletion of variation. We can also start to use that to look at recessive disease genes. So knowledge of phase is really critical. For example, if a recessive disease gene has 2 loss of function variants in it, it really matters whether on the same haplotype or opposite haplotypes. That's the difference between a diagnosis and not. So the high level logic that we had behind this approach is that if 2 variants are in cis in the general population, they're probably in cis in your individual. If they're in trans in the general population, they're probably in trans in your individual. So we did this also on the Nomad V2 data set led by Mike Well and Lauren Francioli. And what we did is actually a 1995 EM algorithm here again. So we can use in this sort of play example here, if we have 2 variants in trans, we make this genotype counts table. We can use the EM to estimate haplotype frequencies, and then we come up with P trans. So it's the likelihood that 2 variants are in trans with each other. Or the probability of inheriting each of the haplotypes that contain only one of the 2 variant allele pairs. So we applied this to the Nomad dataset. We actually did it both overall, so across all of the genetic ancestry groups, and then we did it for every major genetic ancestry group separately. We have these estimates, and we'll have a comparison of the 2 of them in a moment. Just to say, this actually worked pretty well. The reason we have to do this is there's a lot of short read exome data out there, and using proper phasing approaches like ShapeIt5 do not work. We messaged the authors of ShapeIt5, and they were like, no. So if you have short read exome sequencing data, this is where this approach comes into play. When we applied this to about 5,000 trios that were jointly called with Nomad, we removed some individuals to get rid of relateds in our final data set, but they were processed. This is a perfect truth set because we have phasing by transmission, right? We know dad had A and mom had B, and if the child has A and B, we know transmission perfectly. And we found across about 400,000 pairs of variants, we had pretty high accuracy. So there's 5-7% of variants that can't get phased, and it's because both variant pairs weren't present in the Nomad database. We have some that have intermediate values, but of the ones we could phase, our accuracy at the top here, you can see 92 to 99%. So we're doing actually pretty well here. We do see that we're a little bit better at predicting variants in trans, which means if we say 2 variants are in trans with each other, we're pretty confident about that. Variants in cis, we start to struggle a little bit, particularly when you have a super rare variant and a really common one. And that's because we think this is where we struggle the most. We think potentially because the common variants floating around potentially on multiple haplotypes and the rare variants arisen on one of them. So that one's a little bit harder to separate, but I like to be honest about where we work and where we don't. We did also compare these genetic ancestry group specific estimates to the overall one. So the pro of doing all of them together is you have bigger data, you can phase more variant pairs. However, the con is that you might have ancestry mismatches. So we do show actually fairly similar accuracy for variants predicted to be in cis across whether you're using the genetic ancestry group specific one or these kind of high level cosmopolitan ones. However, we do see particularly for African genetic ancestry and East Asian genetic ancestry, a drop in accuracy for variant pairs in trans. So again, the genetic ancestry mismatch there comes into a little bit of a play, but there is a very different number of variants you can phase with these approaches. So our recommendations is to start with a genetic ancestry group specific phase estimate. And if you don't have it, just jump up to the cosmopolitan one. So obviously phasing is impacted, the accuracy is impacted by distance. We both looked at physical distance, space pairs, as well as genetic distance. Again, like in trans, opposite haplotype were pretty good across these distances, but unsurprisingly, cis variants that are going to get split by recombination really impacted by the genetic distance there in sendum organs and physical distance as well. We did also look at recurrent mutations. So CPGs that are methylated are very easily deaminated, and you get a lot of mutations there. They're highly mutable contexts, but actually our accuracy was pretty good for these, even across low, medium, and high levels of methylation in the data set. You can see a little bit lower accuracy here for two variants that are both highly mutable compared to like transversion that are not as mutable, but still like honestly, not bad. I'll take it. Right. So this is what impacts accuracy. We tested this on a gold standard trio set. And the next question is whether this is actually useful in a clinical context. So here as part of the Broad Center for Mendelian Genomics and a large collaborative rare disease data set called GREGOR, we selected a set of patients that had a confident or strong candidate recessive disease diagnosis. So that means that they have two loss of function variants, let's say, or two missense variants in the same gene, and it matches the phenotype. So this is a strong candidate. And the presumption is that these are in trans. They don't always have full trios, so they're not always sure, but these are strong candidates. So we start with 627 variant pairs. About half of them aren't present. Oh, both of the variants aren't present in Nomad, so that's where we lose our biggest amount there. But of the 293 that were both present in Nomad, 96% of them we predicted to be in trans, which matches what we'd have. We had a handful that were predicted in cis, and 5 that were indeterminate. So these 7 here, you'd say, this is where we got it wrong. Well, possibly. When we look into them, 6 of those 7 were proband only, so they actually didn't have the parents. So this is a situation where maybe we're wrong, or maybe they're actually in cis with each other. So we've sent this back. I actually haven't heard whether those got resolved, but one of them were definitely wrong. Had the full trio, had perfect phasing by transmission. We got that incorrect, but overall 96% matching of what we could face, which I really want to emphasize here. Okay. However, finally, this is just these variant pairs. We also wanted to start to do it on a gene level. Can we identify genes that don't have rare in trans loss of function variants, for example? So could we provide a sense of how frequently we're seeing compound heterozygosity in this data set? So we counted individuals that had 2 rare variants in the same gene. We stratified by predicted phase, allele frequency, and consequence. And here we're using some ClinGen recommendations. So loss of function is obviously the most severe, but for missense variants, we're using this Revel and Sierlico deleteriousness score to categorize them into supporting, moderate, and strong. So to summarize this, really, in this data set from like a relatively healthy reference population, we predict very few genes that have compound heterozygous carriers of rare deleterious variants. So the homozygous here is in this blue, and the compound het are in this kind of salmon color. And for rare variants, really not a lot, particularly for loss of function. I should note, massive limitation here. We use Nomad to train this model, and then we tried to like also predict in our own data set. So this is a problem, and we can only really predict in trans variant pairs down to an allele frequency of about 0.5%. Go ahead.
You know that. So it could obviously be that there's an epistatic interaction that takes compound synthesizers they better as we might expect. So if you have two variants that are in the adjacent exon to
Yep.
something, they basically may neutralize the effects of one or the other, right? So
Yeah.
have local epistatic. Ken Kidd showed us a long time ago with detailed analysis that you can do a lot of epistasis within a little or gene.
And they were definitely in trans.
I thought we were looking for.
Yeah, we're looking for compound hits because I know that there's evidence that if a loss of function variant appears on one haplotype, you can get more loss of functions building up. Yeah,
This.
yeah. In cis, yeah, we see that. I think I was telling some of the students earlier, if you have two singleton variants, like they're both only seen once in the data set, we don't provide predictions for them because they get intermediate p-trans values, but 93% of them are in cis. And two rare loss of function variants are almost always in cis. There's both some population genetics data that makes sense for that, but also you don't want like trans loss of functions floating around a healthy population. So of this data set, there were only 28 genes with predicted compound heterozygous loss of function variants, and 25 of those 28 were non-disease genes. So we applied that loss of function curation framework again and ruled out almost all of these. So again, we got it down to 8 loss of function variant pairs and 7 non-disease genes that look like they're both possibly loss of function, loss of function. So again, we saw something interesting. No, we didn't. When you curate, you can really clean up a lot of this evidence. So we have all of these, I should say all of this data, these like variant pairs are on the gene page, and you can look up the predicted phase of 5 billion variant pairs in Nomad V2. We started to move this over to Nomad V4, but the combinatorics with the much larger data set are a thing we're dealing with right now. We're gonna have to work on some computational efficient approaches.
The question, sorry, related to what Rob was saying, it's not clear to me, there's no way to predict, any other gene interaction, this is by gene prediction, right? So that also just.
Yeah.
There's no other way, is that there's any way to predict in spaces.
So I'm a little bit curious about going into gene A versus gene B. However, we have to see if it works with our IRB, because the more you get, that starts to get more identifiable. So we start to run into like some ethical heads up there. It's something we're somewhat curious about, but again, we don't have phenotype or anything like that. So we could be able to say, we have X number of individuals that have a loss of function in gene A and a loss of function in gene B, but it might be hard to do anything else with that if you care about a particular phenotypic outcome.
Just remind me of similar work done in population I in Bradford. There was a work from Kitchener, someone from Sanger, which now is in Texas. And, what? Yeah.
Oh yeah.
Yes.
Yeah.
Did you remember? It was gene sensitive, but the population of Pakistan.
Yep.
Origin, mountain.
Yeah. I know that cohort.
Just remind me of how much the demographic history of the population
Absolutely.
have an impact on that. And
Yep.
there's no way you can predict. I mean, you can model the general ancestral population.
Yeah.
No, it's not here, right? It's not-
Yeah, so yeah, absolutely. We don't see compound heterozygous loss of function variants in our, for lack of a better word, outbred population. But as soon as you start to introduce consanguinity, you do see it. So there have been a number of papers in this space that if you have runs of homozygosity, you're more likely to be able to get, yeah. So the Pakistani population was a perfect example. Yeah.
So you can also speculate about how recent is this, like, if the ransom was a husband is really large.
Yeah.
then some reason.
Yeah.
Sorry, not to, just to say, there's no way you can take into account demography here. Is that also?
I think that,
And also, this is a proof that the population is fairly outbred.
yes, yeah.
It's even the other way. Like,
Yeah.
it's a clear sign that the population is outbred.
Yeah, and around the same time as this, someone did phasing in the UK Biobank, and they found very similar results. Like there just aren't that many disease genes that have compound heterozygous loss of function variants in a relatively healthy population. I think they found one or two new disease associations with this that might not have been found before, but like not a lot. But hey, that's great, right? A relatively healthy population does not have loss of these genes. That's exactly what we expect to see. So it's nice that it's lining up, but it does mean you have to sort of, when you're coming to present it, you're like, hey, we found nothing, but that's good. It's
Well,
this
that's
little,
just selection.
yeah, yeah, love it. It's working. Cool. So this last bit is a little bit of a different thing. This is focusing a little more on rare disease diagnoses. So just to say, we've been getting better and better at finding new disease-associated genes, partially via some of these large sequencing efforts, which highlight genes that are intolerant, and then you can prioritize them in rare disease settings. But even with all of this increased access to testing and increased knowledge about what causes rare disease, still approximately 50% of rare disease patients remain undiagnosed. And often what happens is something like this. A whole genome is done, nothing is found, and then the disease gene is published a month later. And nothing is done again. It's very infrequent that you actually see reanalysis in a clinical setting, especially in a company setting. They're not paid, like insurance doesn't reimburse for that. Frequently people looked at once more time and like, that's about it. So this happens quite a lot. And the number of undiagnosed individuals is increasing in the background. And this is a lot of data that's getting stored in the background. So this is a situation that would really benefit from reanalysis, just something creeping in the background as information builds up. And we know reanalysis helps. When you do reanalysis, this is a meta-analysis across about 30 studies, we do see an increase in diagnostic yield of like 5 to 10%. So you're not solving everyone, but even within a year, like reanalyzing the same cases a year later, you get more diagnoses out of it. However, reanalysis, as I said, insurance doesn't cover it. It's also a lot of time and effort. In the hands of our clinical geneticists, it takes about 30 minutes to 3 hours per case. That's a lot of time. And if you have every year thousands and thousands of undiagnosed cases piling up, this is just not, it's not doable. And reanalysis, unlike kind of initial diagnostic pipelines, has a totally different focus. So here you actually might not want a situation where you're getting 100% sensitivity. So, right, you're getting every single diagnostic variant, but maybe you really want high specificity. Like you don't want to be looking at things that aren't related. So we, in this high specificity set, we want an automated analysis that's not necessarily always going to return a variant, but maybe it does. And the tools that sit out there for automated analysis are a little bit more focused on initial discovery. So actually, a lot of the time, they give you a rank list of variants, and you have to look through 10, 20 of them to find it. So we really want a high specificity approach here that's not going to return something unless it's pretty confident about it. So this is an excellent challenge for like automated, iterative, scalable approach. So as part of a collaborative network, we have worked on this tool called Talos. So in Greek mythology, Talos was a giant that wandered around the island of Crete 3 times a day to protect it. So this is supposed to be an automatic approach where if someone doesn't get a diagnosis on a regular basis, they are reanalyzed with updating databases. So ideally you run it, let's say once a month, knowledge changes quickly. It is deployable in research and clinical settings. Currently, it only handles single nucleotide variants, copy number variants, and structural variants. But it does also look at trios or singletons. This is a collaboration with Daniel MacArthur in Australia, Zornitza Stark also in Australia, the Broad Institute, and Microsoft Research. And a lot of this was done by Kaylee Alquist, who was in my team, and Cass and Matt in CPG in Australia. So the way that this works is that you give it genomic data, you give it some pedigree information, you give it phenotypes, you don't have to, and then it goes and gets both static annotations and these constantly updating dynamic annotations. So these are the gene disease associations that are constantly changing. These are variant associations that are constantly changing. And it does like a pretty straightforward variant logic modules, like, is it a de novo in a gene that's associated with a disease? Like that's one way that you'd prioritize. And just to give you a sense, it's actually pretty cheap. So for 1,000 individuals, we're talking about a couple of cents here to run this. And then at the end, you get these monthly reports, high confidence candidates. And then you can have analysts manually review this. Like, we're not saying this is definitely diagnosing. We're just trying to like ping you when something interesting comes up. So to assess the performance, we use 2 really well-studied research cohorts. These have been combed over a bunch of times. So the first is the acute care genomics. This is coming from Australia. These are really, really critically ill infants and children. So this is the first time they're being assessed, higher prevalence for well-characterized conditions. And the more challenging one is coming from Broad. It's the Rare Genomes Project. Almost all of these families had prior genetic testing. So this is like a gnarly set of cases that have been hard to solve. So this matches also the age distribution and the conditions. So ACG, this acute care genomics, is mostly young individuals with neurodevelopmental disorders, but the Rare Genomes Project is spread across age and has more neurological conditions. And similarly, the trio versus duo or singleton breakdown, a lot of trios for critically ill infants, not so many when you have adults coming to you. And you can also see that in the diagnosis. So about half of these individuals from ACG have a diagnosis that had been found, only about 100 out of 700 for RGP. So these are the data sets we're looking at. And we apply Talos and we say, can you find the diagnoses? And it does for some of them. So on average, this was returning about one variant per trio to look at. That's not a high that's not a high like analysis burden. 86% of in-scope diagnoses were found. In-scope means like if it was a tandem repeat, this doesn't look at tandem repeats, can't hold that against it. This is for ACG, slightly lower diagnostic rate for the RGP cohort, so slightly lower here, 81%. That's because more of the things were kind of hard, complex things going on and a little bit more variants returned. These out-of-scope diagnoses, we are working on pulling in a few other variant classes, so hopefully that will improve. But okay, looks like it's doing a reasonable job on like well-known diagnoses in these cohorts that have been looked at. What about using actually that iterative nature on a prospective cohort? So this is coming from Australia, 47,000 unsolved rare disease cases. Again, a sort of split of phenotypes, a bigger age gap. A lot of them actually only had a limited panel of genes tested before. That's kind of critical. That means that because of their condition, they looked at 20 genes or 50 genes. They didn't look at the whole exome or genome, and a far lower number of trios versus singletons. So again, a little bit more complicated here. And what we find when we run Talos is about a 5.2% diagnostic yield. So about 250 new diagnoses. And this is being split here by the kind of rough reason it was a new diagnosis. So at the top is gene disease associations. A lot of this is coming from new gene disease like associations in the literature. So at the time the person was tested, we didn't know this gene caused a disease. Now we do. The second is variant reclassification. So ClinVar entries have been updated. That's a big aspect here. We also have missed and manual analysis, either from limited panel is a big reason here. And then actually a lot of them hadn't had structural variants or copy number variants called. I will highlight that about 50% of these are coming from new gene or variant information. This is exactly where that iterative aspect matters. And if you break it down by the sub-cohort, we do see different patterns. So a lot of neurodevelopmental disorder diagnoses were coming from the new disease gene associations. Apparently, ClinVar changed some of its cardiac classifications, and that's why this is a major driver here. It was interesting to see that they had slightly different patterns. And unsurprisingly, diagnostic yield was higher for older cohorts than for newer cohorts. Like this is a constantly changing field. There are more disease genes known at this time than at this time. So we do see a range of about 4 to 9% diagnostic yield when we look across the different years. Yeah.
Who ends up using this for PWAS of any cohort?
We have not talked about this, but
Run through all that. You're primarily using exome.
yep. Oh, well, we're using whatever genoid, whatever we have. You need a VCF, you need variant calls. You can use HPOs are used if you have them, but you don't even need them. This is really low hanging fruit though. This is like ClinVar 2-star variants or a de novo in a particular gene.
Is there anybody else who's run it through a Generon RGC or UK Biobank and say, what do you see for associated for pleiotropic patterns?
No, we haven't yet. This is still a preprint. Like this is this is new. We're still like playing with it. Yeah. The goal was to get a version 1 out that was like the setup is like we can have it running monthly in the background, just be pinging people when something new comes up. But you're right, there's obvious useful expansions here. Right. So this case we talked about earlier, a whole genome done in April 2022. The genes published, a month later. As soon as they enter reanalysis, they're diagnosed because the knowledge is known. So that's the vast majority of those 250 diagnoses, but 6% were found via iterative cycles. So this case, for example, whole genome done in 2020, they entered reanalysis in July 2023, a couple cycles ran, and then this gene was changed to a strong association for autosomal dominant, and they got diagnosed a month or two later. And we see an average of about a month in between new knowledge being revealed and a Talos diagnosis, which makes sense because we run it on a monthly basis. This is not a huge analysis burden. It's about 5 new variants per 1,000 cases when you're in an iterative cycle. Again, low-hanging fruit. We're not giving it, it's high specificity. We're really only pushing forward stuff that we think is probably going to be a diagnosis. So this is on medRxiv. And we have a GitHub that's uses Nextflow and is fairly minimal. We're happy for people to play around with this right now. And I should say this was a big collaborative effort with a lot of this coming from CPG in Australia and help from Microsoft as well. Yeah.
All cases that got diagnosis and see if they had any like medical intervention.
I think there were medical interventions for some of them. I didn't show it. One of the other ones of this figure is the RNU42. We found a bunch of those, which, yeah. Yeah, I don't know. Zornitsa might know if some of the ones from Australia have some interventions, but not as you know fairly well, not all rare disease diagnoses come with some sort of therapeutic or medical change, but some of them do, and it can be really impactful. So I know from our work anecdotally this happens a lot, but I don't know the rates in this particular cohort. Right. That's it. Just to say Nomad is going to get bigger. We're pulling in all of us. So our next release, hopefully next year, is going to have 440,000 genomes that have been processed. And after that, we're doing a federated approach. So for data sets that can't leave their country for very good reasons, we're working with teams from around the world to process data in an equivalent manner. And the goal is that they will share aggregate allele frequency back with us. We will support potentially spinning up their own browsers for their particular data set. And this will increase ancestral representation in our cohort. So Australia is one of the first ones that will be on board. Singapore has a data set that's pretty close to ready to go. Yeah, so this is with Nikki Mulder. So I think it's... I don't know if it's H3 Africa. I would have to check on our website, but there's a cohort there. Yeah, that's why I don't want to, I'm pretty sure it's not H3 Africa, but it's another one of them. But the goal is if we can show a couple of examples of how this works that other people. No, not necessarily. I think it's large collections across the continent. Yeah, but you make a good point. There's places in the world we know we need to do better. And East and North. Well, yeah, I should have said I had South Africa on there, but I think it's not only South African individuals. So yeah. Anyway, so we've talked about variant curation, selective constraint, statistical phasing, some automated reanalysis. There are a lot of people to thank. Obviously, Nomad has a ton of people involved with it. I've just listed the core team members, but all of the individuals that contribute, including the participants as well as a lot of the analysts. I've listed some of the Talos authors and our funding. Thank you very much. We're a little over, so we can go into, I know we have extra time right now. Yeah.
I love your simple slide that was just one sentence that is, if it looks too good, it's too good, essentially. Kaitlin and I went through this when we sequenced our first mouse strain.
Yeah.
We found all kinds of wonderful, crazy variants, and nope.
Yeah, I actually think Daniel McCarthy had it in a paper a while ago, something along the same things. Like this is how I treat science, though. Like my entire career, this type of idea has gotten across. Like if you see something interesting, you really have to prove that it's not. And only then, maybe, can you get it across the line.
In this context, that is the fitness landscape is very important. Anywhere away from where you are, the arguments work well. So then any error is making
right
interesting. So
yeah, well, that's why we wanted. You need to eliminate error because the error is going to show up in the places.
this back in the day, I think maybe it's a little bit related to evolution,
Okay.
not natural thing inside. Like
Ah.
dropping on the system, it's kind of like, we see it at this state of optimality.
Okay.
So that any fuzz on top of it is very pushing forward. Wow.
Okay, I guess I'm thinking mostly from the perspective of if you want to assign a new disease gene, you better prove that it's not an error. It's not some sequencing error before you go spend a whole bunch of money making a mouse model to try to understand the molecular mechanisms behind it. So that's maybe where I'm coming from, but I can see, yeah.
When
Yeah.
we did it, our main problem was gene model error.
Mm.
So, we had something that was,
Yeah.
exon, it turned out it was 3' UTR or something like that.
Okay, yeah, right.
Yes, leads to a question.
Mhm.
So are you concerned that you don't have a complete model and transcript page? That would seem to be really important for a lot of the things and especially a lot of something that.
Yeah, we are limited by the transcript annotations that are put out. And certainly in some of these constraint scores, we've run into issues where the transcript that's been selected is malformed, or it's not actually a good representative. And since we do these things at scale, we can't always necessarily go in and handhold every single thing. So there are, well, fine, I'll use an example. CSMD1, if you look it up in Nomad, it has a synonymous score, meaning like we're predicted synonymous variants and compared of like 11 or something, which means that we are not predicting synonymous variants well in that gene, which means I don't trust our missense or loss of function predictions, but it's also a paralog. There's CSMD2 and CSMD3. Probably this is a mapping issue. We have not been able to handhold and figure out exactly why that one's going wrong, but it is a disease-associated gene, so we should. There's a handful of other ones in prior versions of Ensemble where the canonical transcript that we tend to use as the representative was missing a methionine at the beginning or a stop at the end, and so we just didn't process it either because we said it looks bad. You're right.
And then there's non-canonical transcript, which you have basically, huge grammar of the possible.
So we
Talk
have
about modeling on space directly is like the transcripts, the functional annotation you have is just so much smaller than the genomic data that you have. You're always in this place where.
So we have run these gene level constraint scores on V2. We ran them on every transcript that was available. It's like an extra file. We only usually analyze one representative, but we have it for everything. They tend to be fairly correlated, but you're right. There's some, I had always been interested in whether you could like use this information as a way to identify a transcript that seems like it's not actually that relevant because it has a very different pattern and profile here.
Some of those loss of functions are survivable.
Yeah. Oh, we have evidence of that. That's one of the things part of the curation framework is they use this score called PEXT. It's a proportion spliced in. It's using GTEx data, but you can see if a particular exon is only in, 20% instead of 100%, that's where you see loss of functions. MEF2C actually is a great example here. There's a handful of loss of function variants in MEF2C, but they're in exons that are not spliced in all the time. And so it looks like that's probably why they're tolerated. Otherwise, we wouldn't have expected to see them. Yeah. Yeah. So we do take that into account in some way.
It's more sparse.
Yeah. No, good questions though. Oh, do any of the trainees have questions too? I mean, yeah.
So, I know the Nomad, you have the catalog available for the Nomad.
Yep.
You know where there are plans to just do genome-wide catalog to make the data available? I know
Oh,
it's crappy.
No, it's just so expensive. It's so expensive. We're working on expanding it out, but it's very expensive. Yeah. So Ben Weisbird, I think, has some plans to do it on maybe a subset, but I don't want to promise, particularly in this funding environment, like we're trying to make sure that we're doing the most important deliverables. Yeah.
Within scope.
Yep.
Why were they missed?
Great question. Yeah, no, we have a whole breakdown of some of them. So for a few of them, it's because it was like one of the two, let's say a compound heterozygous, one of the two of them is like not as convincing. So an analyst, like a human looking at it, would be like, this is a very convincing loss of function, and this missense is on the border, but the phenotype matches really well. So I'm going to use my clinical judgment to push it across the line. But our very dumb computational approach that's very conservative says no. So a lot of it is because we're conservative. Yeah. Or read quality is a big one. Our curators will go rescue things that have really bad, like they look really bad read quality, or any of these quality control metrics. But if it's a compelling enough candidate, they'll like do a dive into it to rescue it. But again, a like high level computational approach will say, nope, I don't trust that variant. Yeah.
My misformed question about trans and cis, we're gonna flip.
Yeah.
But I would like to come back to the
Sure.
topic of stasis as a general theme, because it's really puzzling me more and more as the sample size we have for mouse genetics gets larger. We see huge amounts of epistasis or very, very complex trait mortality, basically. And I'm trying to wrap my head around why people like Trudy Mackay and Jim Shepard, for that matter,
Yeah.
really hammer on the importance of epistasis. And if you go back to the initial rare variants, A, B, and C, non-agouti, black
Yep.
and albino,
Yep.
it's all about epistatic interactions where albino basically clobbers everybody. But in human genetics,
We've not seen a lot of evidence of it.
we
Yeah.
don't even talk about
Well,
it. And I understand
yeah.
the reason. We do have large sample sizes, obviously, unrivaled
Yeah.
large sample sizes. But we also have this genetic structure where you're looking at 10 to 50K blocks. So you can't really afford to do a genome-wide,
Well, Rob, there are cases, big runs of homozygosity.
Yeah.
Yeah.
So those,
Yeah.
I'm just looking at those, yeah.
So you introduced the topic of G to P.
Yep.
And I guess, where would you expect us to be in 20 years?
Oh.
Moving G to P, and is it all gonna be additive effect? Or are we gonna grow up and do epistasis, at least two-way epistasis?
Ah, man, are we gonna grow up? Wow, with the gauntlet thrown like that. So the tricky thing is a lot of the literature and looking at the human results are seeing mostly additive effects. You could argue some of it's a power issue still, but
Only one method that's been applied. At
Yeah.
Scale anyway. And it's on small number of markets. It needs to be done. So
it could just be that we need to just do it. Possibly in 20 years, maybe we will have grown up and done that. But I think the epistasis has been hard to show.
probably better models of interaction
Yep.
that you can use to shape those queries.
Yeah, so I've been a little bit interested in trying to do these sort of like rare burden approaches focused more on protein complexes themselves, where you'd anticipate the interactions to come up. I'm working, it's like goals to put this in a grant, but like I have to work through the power calculations to see if we're actually able to do it. Because there's databases out there about digenic inheritance, right? Mutation in one gene, mutation in another gene, that's how you get disease. Most of those are bad. Most of those don't actually hold up when you look through the data. This is compounded by the fact that incomplete penetrance is obviously a thing that we run into, but there's some of them that you can see in a totally healthy person. There are a handful of them that seem semi-convincing, maybe. But the tricky thing in this space is any two rare variants in two different genes, the likelihood of seeing them again in a randomly healthy person is super low. So we have to get a little bit more. Anyway, we've tried. We've looked at this database. Some of them we're able to rule out. Some of them we can't rule out, but we think are suspicious. So we're thinking about it. Yeah.
Just stay mystified for the next 20 years.
Hopefully not that long. I mean, really what we're limited by here is we have some of the sample sizes, but it's not as controlled of an environment, right? So you need to I actually think we need more data.
But if I give you mouse epistatic comparisons, comfortable about it.
We might be able to check.
Then it's strain model test.
Yeah, no, we might be able to check if we had some very specific things that we were looking at. And that's why the thought process was to do directly interacting things, because it's more likely to be that way. But again, you lose power really quickly. And this is totally a statistical power game to be able to nominate candidates in human genetics. But then maybe you can go test in a model system or in a cell culture to say, hey, these, like we see this impact of these two variants in these different genes. I thought there was some work, Duke, Nico Katsanis doing this a little bit and under the 16p1 1.2 duplication and deletion about two genes there that might have had some epistatic interactions, but yeah, very few kind of bona fide examples. Yeah. Okay. Well, sharing is caring. All of our data is public on the internet because that's how you should do it. I say as someone that has this large data set that we put on the internet, so easy for us to say.
What we want to do with it. We
No.
want to take things like Nomad and project them on.
Oh, yep. So we've had some chats about this. It's
Not alignment to a graph.
right.
I mean, you take the reads.
Yeah.
And match them to the haplotypes sequences. It's a better, like if you do that, it's kind of mathematically. Unique what you get, and then you can reprocess it later. You make different alignments or representations later.
Okay, yeah, I guess I should say our crams aren't available. Yeah. I said, I guess I should, our crams aren't available. Our read data is not available because that's personally identifiable and thus blah, 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 blah. Yeah.
Yeah, but I think about talking.
We're happy to discuss some of this. I think some of it would be, since this is like publicly facing and goes into clinic, some of the graph representations that is hard for people to understand. So we still need a standard representation.
Matches across the tangent sequences.
Okay.
Like I've seen this sequence in this.
Yep.
Position. So.
Okay. Yeah.
You have some time.
All right. Other questions, concerns?
Ready?
All right. Thank you.
St. Jude Marathon tomorrow. Yes. And then there'll be kind of a party, right, very close to us, I think, in the, whatever this neighborhood is.
Oh, yeah, there's all kinds of places.
Anybody running? 1, 2, 3, wow. You're not running?
Nope. Not a runner. Not a runner.